Hi everyone, welcome to my channel. So in this video we'll be talking about hemoglobin. We have just completed composition of blood and started blood physiology. So this is our second lecture in which we'll be talking about hemoglobin, some aspect of hemoglobin for your theory paper. So what is hemoglobin? Hemoglobin, it is basically made up of two words, heme plus globin. Heme, what is heme will be doing and what is globin will be doing that, okay. But overall, what is hemoglobin? It is a red oxygen carrying pigment in RBCs which consists of heme plus globin. So let us see what is heme first. So what is heme? Heme is basically iron containing porphyrin. What is porphyrin? Porphyrin is basically 4 pyrrole rings, okay, tetrapyrrole, because there are 4 pyrrole rings. So 4 pyrrole rings, they are joined by 4 methane bridges. So to understand structure of hemoglobin, this thing is very important, that what is heme basically, okay. So this heme having 4 pyrrole rings joined by methane bridges. What that mean to you? So let us see. So these are the 4 rings I have drawn. These are the pyrrole rings and I just joined with methane bridges. All the rings are joined. Okay. So these are methane bridges and these are joined. So whatever carbon is left here, I just numbered it 1, 2, then leave these 2, then 3, 4, again leave these 2, 5, 6, again leave these 2, 7, 8. Now, are you getting me how I have drawn this structure? 4 pyrrole ring, base is toward center, right? And methane bridges are just joining them. Okay. Next is 4 pyrrole ring are joined by methane bridges. Next, what is next step to draw hemoglobin structure? Methyl group you have to attach at 1, 3, 5 and 8. Okay. So we have already numbered it. So let us attach methyl group at 1, 3, 5 and 8. So let's do that. So methyl at 1, okay, methyl at 3, methyl at 5, okay, and methyl at 8. Okay, so first thing we have done. So this is done. Okay, next step is 2 and 4 position. We have vinyl group. So where are 2 and 4 position? This is the 2 position and this is 4th position. So let us attach vinyl group. Okay, again here vinyl group. Fine, this is done. Okay, so this step is also done. Next is 6, 7. At 6th and 7th position, we have to attach propionic acid. So where is 6th and 7th? This is 6th and this is 7th position. So let us do it with 7th first. So here what you have to do is propionic acid, CH2, CH2 and acid, so COH. The same thing we have to attach here, CH2, CH2 and COOH. Okay, so these 3 steps are just completing your hemoglobin structure, where you have to just put iron compound tightly bound to this hemoglobin structure. So are you getting me? Now we have drawn hemoglobin structure very easily. I hope this point is clear to you. So this is the complete structure in which these are 4 pyrrole rings. Okay, at 1, 3, 5 and 8 position we have methyl, we have vinyl at 2 and 4 and we have 6th and 7th we have propionic acid. So this is complete hemoglobin structure, basically heme structure. So let us talk about globin. So globin, what is globin? It is a protein component. It consists of 4 polypeptide chain. Okay, so 2 alpha chains having 141 amino acids, 2 beta chains having 146 amino acids. So 2 alpha and 2 beta chains are present in one hemoglobin structure, and each chain either 1 alpha or 1 beta. So every chain it attaches with one heme. So 4 heme groups are there with 4 polypeptides. It basically make one hemoglobin. So 4 proteins, 4 chains and 4 heme group. One hemoglobin have 4 iron attached to 4 molecules of oxygen. One iron basically attached with one oxygen molecule. So one hemoglobin is having 4 chain and 4 heme. So 4 iron, and 4 iron basically it combined with 4 molecules of oxygen. I hope now the hemoglobin structure is clear to you. Now let us see varieties of hemoglobin. So we have HbA, 95% we have HbA, okay, 2 alpha and 2 beta chains. HbA is the predominant type of hemoglobin present in adults. So if after birth HbA is not formed, HbF will persist, causing thalassemia. Okay, so we'll do it in later videos. So HbA is basically the adult hemoglobin which we are having. HbA2 is nothing but when beta chain is replaced by delta chain, there are 10 amino acids which are different. So HbA2. What is HbF or fetal hemoglobin, which is present in fetus till 5 months? Okay, where we have 2 alpha and 2 gamma chains are present, where 37 amino acids different from HbA. It's high in intrauterine duration and at birth it reduces. 90% is HbA at birth. Okay, HbF has great affinity for oxygen. If it persists after birth, it's thalassemia. Then last is HbS, which is sickle cell hemoglobin. In beta chain at position 6th, glutamic acid is replaced by valine. Okay, in reduced form, when it's not oxygenated, this HbS, it precipitates into crystals and damage cell membrane, which leads to viscosity increase, and these hemoglobin, these RBCs are destroyed by spleen. Otherwise, hemoglobin can exist in various forms in our blood. So oxyhemoglobin when hemoglobin is combined with oxygen, deoxyhemoglobin when hemoglobin is without oxygen, methemoglobin when iron, this ferric form is present instead of ferrous form. Okay, carbamine hemoglobin when it's combined with CO2, carbonyl hemoglobin when it's combined with carbon monoxide, and glycosylated hemoglobin, which is HbA1c. Okay, so whenever hemoglobin is basically destroyed in our blood during lysis of our cell, hemoglobin is converted into heme and globin. Globin goes to amino acid pool, which is reused for hemoglobin synthesis, and heme is converted to biliverdin, which further is converted to bilirubin by liver. So in liver, conjugation of bilirubin is done, and which is then secreted in urine as urobilinogen and feces in stercobilinogen. Okay, so this is all about fate of hemoglobin. Last thing is the normal values. So in adults we have 14 to 18 gram per deciliter for males and 12 to 16 gram per deciliter for females. At birth we have 17 to 22 gram per deciliter, and in children's or infants we have 11 to 13 gram per deciliter. So this is all about hemoglobin. This can be asked as question, like you can be asked about structure of hemoglobin and fate of hemoglobin, which can come in your exam. So I hope the point is clear. Next video will be talking about erythropoiesis. Thanks for watching, guys. If you want Hindi videos, you can visit my Physiology Hacks channel. Thanks for watching. Good luck.